So this video is all about the process of transcription. So we're going to be focusing on how RNA is actually being made. So if you remember, the process of transcription is when I go from DNA to RNA. Since my DNA is inside the nucleus, this would make sense that the process of transcription is also happening inside the nucleus. So if I'm looking at the central dogma, I'm going to be focusing on this section, the process of going from DNA to RNA. So transcription, once again, is happening inside the nucleus. Once we have our RNA, then the second step is going to be from going from our RNA to actually making our proteins. But today we're focusing on this section. So just to recap, we know that there are 3 different types of RNA that can be formed. We've got your mRNA, which stands for messenger RNA. It's pointing to this long single strand here. Your rRNA is this large glob here, which stands for ribosomes. You'll see it's labeled as a ribosome. And our third type is called tRNA. It kind of looks like it's shaped like a T. It stands for transfer RNA. So we'll talk about those 3 types in a little more detail. So mRNA, it's called mRNA because it's called messenger RNA. And what it's doing is it's taking the message from the DNA. So we know that DNA is the instructions or the code for ultimately making the protein. So if DNA has the original code, mRNA is going to basically take that message in the form of RNA. So it's a single strand of RNA. So if I look at my model here, this is my single strand here of RNA. I know it's RNA because I see uracil instead of thymine. And basically I'm just rewriting my code from DNA, and I'm going to have uracil instead of thymine. This is going to serve as our template or something to build off of. So we're going to take all these sequence of letters, my A's, U's, G's, A's, C's, A's, and I'm going to use this sequence of letters to actually build our proteins. And if you remember, our proteins are made up of lots of amino acids all linked together. The second type of RNA that can be made in transcription is rRNA, and the R stands for ribosomal RNA. So we take that same single strand, but you kind of fold it into this large globular form. So if I look in the picture here, I'm looking at this large kind of orange glob here. Another name for rRNA is basically a ribosome. Okay, so if you think back to our cell city unit when we learned about the cell organelles, what are ribosomes responsible for making? They're responsible for actually making the proteins in your body, and that's what they're going to help us assemble. So the third type of RNA is called tRNA, and the T stands for transfer. Once again, it's the same kind of RNA, so it's a single strand of RNA, but it's been folded back onto itself into a shape that's called like a hairpin. So this funky little T shape, okay, is called a hairpin. So anytime you see these kind of T-like shaped figures, these are probably tRNAs. It's called transfer RNA because it's transferring an amino acid to the site where we're actually going to make the proteins. So if you'll notice, they're carrying these little circles, and those are different kinds of amino acids. There are 20 different kinds of tRNAs because there are 20 different kinds of amino acids. So for every different amino acid that we have here, you're going to have a different tRNA that carries it. And you'll learn more about all the different kinds of amino acids. Don't worry, you don't have to memorize all of the 20 different amino acids. Alright, so let's talk about how we actually make this RNA. So transcription is the process of making these types of RNA, and you're going to use the DNA sequence to actually make the RNA. Okay? When you transcribe something, it literally means to rewrite something, or you're writing something down. So you're taking the code from DNA and you're rewriting it very similarly, but just slightly differently in the form of RNA. So DNA is going to have A's, T's, G's, and C's. When I rewrite it in the form of RNA, we're going to have A's, U's, G's, and C's instead. So it's just slightly different. It's like if you were to transcribe a novel, if you were to go from English into Spanish, it's basically saying the same thing, but you're writing it down just slightly differently. Now remember, the DNA is inside the nucleus, so if I'm going from DNA to RNA, this better be happening inside the nucleus of a eukaryotic cell. So I'm going to go through the process of transcription. Looking at this diagram, you might go, Mrs. Barnum, this looks kind of familiar. It is. There are very many similarities between transcription and replication. DNA replication is when you're making copies of DNA. Well, in transcription, we're not making DNA, but we're going to be making RNA. So yes, there are going to be some similarities to this. Alright, so let's take a look at this. This blue strand here, I know that this is my DNA. I can tell because I've got a double strand. I've got my double helix here. And if I look in my model here, this red strand here is my RNA. I can tell because it's single-stranded. So first what's going to happen is we're going to have the enzyme RNA polymerase. And this is going to attach to just the section of DNA that is going to code for the trait or protein that we want. Remember, there are many different genes on one strand of DNA. We're only making one particular trait. So I'm only looking at one section of the DNA. We're not going to do the whole thing. This is why RNA tends to be a lot shorter than the whole strand of DNA. In DNA replication, it was helicase, but it's RNA polymerase. So that is this kind of green circle here. So that attaches to the site where we want to make this protein. And it's responsible, it's going to break those bonds, okay, kind of like the helicase did, and kind of unwind that helix. This site that it's attaching to is called the promoter region. Alright, what happens next? So we've kind of separated the helix, and now our RNA polymerase, okay, is going to bring in RNA bases or RNA nucleotides. If I were making DNA, we'd have DNA polymerase. We're not making DNA, so DNA polymerase better not come in here. So we're going to have RNA polymerase, so the same thing is going to help bring in our RNA nucleotides. Okay? Now, here what ends up happening. If I see, let's say, this is an A, let's see what I said, this is A, T, A, G, and C. If I were to bring in RNA bases, then I would have, instead of thymine, uracil would bind in, then I've got adenine would bind in, uracil again, cytosine, and guanine. So we're just going to be bringing in my RNA nucleotides instead of DNA nucleotides. At the same time, it's bringing in my sugar phosphate backbone, but it's a different kind of sugar. So it's not deoxyribose, but it's a ribose sugar. We call this the elongation step. In other words, the fact that I'm adding on my RNA nucleotides, so I'm building complementary RNAs called to the elongation step. To elongate means to make longer. So remember, we're only transcribing just a small section of the DNA for the one protein or one trait that we're looking at. So this process of transcription is going to stop when it reaches a termination code, wherever that ends up being on our sequence. Once it reads that termination code and says we're done making our protein, the RNA is going to be released, so you kind of see it like leaving, okay? And then the DNA is going to close back up onto itself and basically end transcription. Sometimes this termination code is referred to as a stop code or stop codon. So if you remember, this whole process is happening inside the nucleus. Once this is done, the RNA is eventually going to leave our nucleus and enter out into the cytoplasm. So just to recap, all three forms of RNA are made or transcribed in this fashion inside the nucleus. So whether it's mRNA, rRNA, or tRNA, they're all made this way, And then if they're folded into a big glob, you get RNA. Or if you fold it into a hairpin, then you get your tRNA. But they're all transcribed in this process. Now, it says this original model of RNA is then going to be processed and some things are going to be done to it. If you remember, on page 221 of your reading, we talked about RNA splicing and these exons and these introns and spliceosomes and all this stuff. So this is talking about post-transcription modification. This is a level 4, which I will go into more detail in a different video. So this concludes the video on transcription. I hope this was helpful.